But the last argument we're going to go over is my personal favorite because I'm a nerd. So we're going to talk about the initial arrival of life and what explains it. By talking about the kind of life that we see and what we learned about it from the fossil record and modern science. So following what we've seen so far, the universe had a beginning. There are implications there's a cause beyond the universe. The kind of universe that was created is very finely tuned for life to exist. Okay, that also has implications. But if there truly is this purpose, we should continue to see this continuing into the arrival of life. Now, what do we find? What kind of world are we living in? Well, you might say that if life arose in the absence of intelligence as merely the product of natural processes, we'd expect it to rise somewhat slowly and start simply. I think we all know that nature doesn't create complexity very quickly. It happens over a long period of time as the standard materialistic, naturalistic, and evolutionary theories. But if life arose as the product of intelligence, we'd expect a more deliberate act of creation. And in this case, we'd find complexity rising quite rapidly and explosively on the scene. That's what intelligence does. That's the mark of intelligence. Complexity arising spontaneously as a deliberate act of creation. These are very different scenarios. What do we actually find in the fossil record? What we find is that the first organisms in the fossil record were actually highly complex, with structures like spines, eyes, digestive systems, and organs. Not simple organisms. What do these look like? We have a couple here. Meet my friends, Opabinia, Marella splendens, Anomelocaris, and Hallucigenia sparsa. Say that last one with me. Hallucigenia sparsa. These guys are cute. I want one. Are these simple life forms? No, you can see complex structures. You see eyes, you see limbs, you see spines. There are digestive systems inside these. These are not simple life forms. But the problem gets even worse. You see, they rise quite rapidly on the scene. It's been calculated that if life on planet Earth were divided into a 24-hour clock, the history of life on Earth, we'd find 20 of the 26 major animal types arising in just 1 minute of that 24-hour clock. In other words, it's just saying that a great diversity arises on the scene very rapidly in the history of life on planet Earth. So what have we found so far? Life arrives quite rapidly on the scene, already complex and diverse. And there are implications of this. This is being talked about and admitted by scientists, as explained by geologist Sir Roderick Impey Murchison. The earliest signs of living things, announcing as they do a high complexity of organization, entirely excludes the hypothesis of a transmutation from lower to higher grades of being. These are the first organisms, and they're already complex. The fossil record doesn't show them coming from anything below them that's simpler. We saw that these are incredibly complex organisms. To say that these came about by natural processes, these being generation one of life on planet Earth, is like saying a tornado went through a junkyard and put together an aircraft. Does natural processes, do they put together complexity of this degree? But it's actually worse than that. It's like saying a tornado went through a junkyard and created an airplane, a tank, a muscle car, a motorcycle, and a helicopter, because we have diversity from the start. Are natural processes capable of that? Now, you might say, if this is the case, then how do people assume that life is the product of natural processes? Why is that even taken seriously? Why is it so mainstream? Well, a lot of it has to do with presuppositions and worldview that gets taken into the discussion. But basically, if someone wants life to be the product of merely naturalistic processes, then you're going to assume that happened, and then you're going to group the data into a format that reflects that. So one is going to assume that these aren't the first organisms, because that would be inexplicable to their theory. In other words, they're assuming that there were ancestors. These aren't actually the first ones. These are much later. These are descendants. In other words, they're looking for ancestral precursors. Now, like I said earlier, these are the first ones we find. We don't have ancestral precursors. So then how do people do this? Well, they give what is called an artifact theory. Basically a fancy way of saying it's an excuse for why we probably shouldn't expect to find these fossilized ancestors. They give reasons for why they're not there, and maybe we shouldn't expect them to be there to begin with. And just a brief thing, there's no such thing as unavailable evidence. Evidence can be defined as the available information or data pointing to a belief or proposition being true or accurate. To say that there's evidence out there that we can't find or shouldn't expect to find seems to be a contradiction in terms, but that's another discussion. Let's just talk about 4 artifact theories. Some say that due to tectonic movement, perhaps these fossils did exist, but they're displaced into the sea, which is why we don't find them below the other layer fossils that we actually do have. But after decades of drilling in the sea and looking for them, we still haven't found any, so many are doubting this theory. Next, some say that perhaps these ancestors were too small to be fossilized. But size doesn't appear to be an issue. We even have fossilized bacteria. And second, you'd expect to find progression. It doesn't go small to large in one generation. You should expect to find some ancestors, but we don't. This one is doubted as well. Third, some might say that perhaps the ancestors were too soft to be fossilized. Okay, that might make sense. Actually, it doesn't. We even have fossilized algae. Further, it turns out that 95% of our fossilized specimens actually are soft-bodied or at least thin-skeleton. So softness doesn't appear to be an issue for fossilization. And lastly, you'd expect to find progression once again. It doesn't go soft to hard in one generation. You'd expect to find some fossilized organisms with some hard body parts, but we don't. And lastly, some say that perhaps the sedimentation of the area was actually not ideal for fossil preservation. This is one of the main ones held to. But actually, geologists have determined that the sedimentation of the area was even more ideal for fossil preservation. What does that mean? We should expect to find even more fossilized organisms of this era than of the descendants we do have. What do we find? It's just the opposite. We don't find any. You expect to find more than what we have, but we don't find any. So this one is doubted as well. So because these theories don't hold up, there's no reason to think that these fossilized organisms existed. And if that's the case, then these organisms we saw a minute ago truly are generation one of what we find in the fossil record of life on planet Earth. And this is a problem that's been run into that's admitted by many paleontologists, as explained by Niles Eldredge. When we do see the introduction of evolutionary novelty, it usually shows up with a bang, and often with no firm evidence the fossils did not evolve elsewhere. Evolution cannot forever be going on somewhere else. Yet that's how the fossil record has struck many a forlorn paleontologist looking to learn something about evolution. So what is the picture of life that we find? Well, take a look at this. This is an oversimplified version of a tree of life. You see here, in the branches you find fossilized organisms that we do find, diversification happening within those major types. But where they allegedly combine, we have what is called a missing link. Now, based on what we saw, there's probably good reason to think that those links didn't exist. If they did, we expect to find them, and in even greater numbers in the fossils than what we do have of the alleged descendants. But what happens if we just hold on to what we do have? What's the picture of life's history that we do find? That look more like this. See, we would find life coming into existence already complex and diverse, isolated within major kinds. And then, of course, diversification does happen within those kinds. That's the picture we get. Life arrives complex and diverse on the scene quite rapidly. But what would it take for life to all be connected? I think this is an important question because the science that's been done because of this question actually reveals the kind of life that we have on planet Earth. It reveals the complexity in the systems and structures and information within life. That's what we're asking next. What does it take to make a new animal? Now, scientists have identified 3 components needed for new body plans. First, new functional protein folds. Now, a protein fold is a combination of about 300 amino acids bonded just right. You might think of it kind of like origami. They're folded just right, making a particular function possible. Now, what are the odds of getting it bonded just right in order to make any function possible? Well, it's like a little molecular machine. In fact, it's been calculated that we only have one chance in 100,000 trillion. It's not just a random glob of goo. It's bonded just right, like a little molecular machine. Now, what's that look like? Molecular biologist Douglas Axe calculated that the probability of generating just one new functional protein fold from an already existent functional protein fold, by the way, that's a generous assumption that one already exists, or the odds of getting a new one from that pre-existing one. He said that if all organisms to have ever lived on planet Earth generated one new sequence, tried one new fold, only 1/10 trillionth of sequences would have been tested. And that's just for one new functional protein fold. The fact is that we actually need whole new systems arising together for a new functional body plan. There's no hope in the history of life on planet Earth to get just one, but how much more whole systems? And this is exactly what we have to assume happened repeatedly in the history of life on planet Earth to get vastly new body plans with new functions. So new functional protein folds as a new component seems unlikely. Second, new genetic information. This is the second component that is needed for a new body plan. Now, to unpack this, let's explain what we mean by genetic information. Genetic information holds what theorists call specified complexity. Let's unpack that term, what that means. First, let's start with complexity. A simple sequence is subject to redundancy and patterns. Now, it's actually that redundancy and patterns that keeps it from being able to hold higher complex information. So a complex sequence, on the other hand, is not subject to that redundancy and patterns, making something higher and more significant possible. but that doesn't mean that's what you're going to find. That's where the specified part comes in, because you see, unspecified complexity, or what you might call mere complexity, it has no particular arrangement, no particular parts, and there's not going to be a particular function. In other words, it's just kind of gibberish. But the specified part says that you have the specific arrangement of specific parts resulting in specific function. This is kind of what we find in an English sentence. In the sentence, you see a word like his. It's not just a random collection of things. In order for there to be the function, you need the specific collection of the specific letters in the specific order and placement. That's specified complexity. And that's exactly what we find in DNA. DNA has genetic letters arranged just right together, making the function possible. Now the question is, can you get new genetic information, new specified complexity in the genetic code from mutation? Now mutation has a couple ways that it can happen. You can have inversions, you can have insertions, you can have deletions, a couple different tools. But just for the sake of simplicity, let's just say, in this sentence, what happens when you just start randomly changing things? Just one letter at a time. Are you getting new information, new function, a new meaning, a new sentence? No, you see, randomness corrupts order. The function was lost. And that's what we find, that information is lost when you try to corrupt the specificity. Specified complexity, the specificity is crucial for the function. Now, randomness is the opposite of specificity. So when you start randomly changing specificity, you don't get new specificity, you get less specificity, you get less information. We see that random changes don't increase order and add specificity and information, but rather they corrupt order and destroy specificity. Now, what's that even look like in the context of life? We see that degraded order results in degraded function. When we start corrupting it, it's going to impact the function on the other end. So you're going to have degraded function. Now, when you have degraded function, you have a loss of fitness within the organism. Now, if you know something about natural selection, survival of the fittest, if you have a loss in fitness, you're not going to go on and propagate your genes and create a new whole type of organism. You're actually going to die out. It's going to cut it off there. So ironically, natural selection actually puts a limit on the amount of mutation that can happen within the genetic code. As explained by Stephen C. Meyer, since genes, like English sentences, contain sequence-specific functional information, multiple changes in the genetic text will inevitably degrade function or fitness long before a new functional sequence will arise. Just as random changes in a meaningful English sentence will typically destroy meaning long before such changes produce a significantly different meaningful sentence. You might imagine that after maybe a lot of changes, maybe you get some amount of small regions of order as a result of random change. But that's exactly what time doesn't allow you, because as soon as you start losing order, life is going to end there. You don't have time to just keep on messing with it, because life needs that function in order to survive. So new genetic information is also doubtful as a result of mutation. But the last component is new epigenetic information. Now, epi just means beyond. So epigenetic information is information beyond the genes. A couple of examples include cytoskeletal arrays, microtubules, centrosome, membrane patterns, membrane targets, ion channels, and the sugar code, just to name a few. Now, what do those do? What does that mean? Do we have any Lego fans in here? Yeah, I know there's a big push for Legos right now. I did Legos as a kid, but then, I don't know, they're so expensive, It's plastic. I don't get it. What's with that? But you might think of new epigenetic information as kind of like a Lego instruction manual. It tells you how to put the pieces together into a higher complex structure. For example, take one of these boards. Just having the pieces, does that tell you how to put it together into the higher complex structure, accomplishing a function? No, you actually need information beyond the circuit board in order to put it together. Or take a house, for example. You need more than just the raw materials. You need more than just pavement and glass and concrete in order to build a house. Instead, you need more information. You need a blueprint in order to put it together into this higher complex structure. That's what epigenetics do. They collect proteins into cell types into tissues, tissues into organs, and organs and tissues into body plans. Now the question then is, can we get new epigenetic information via mutation? Now it's doubtful for two reasons. First, these structures are really too large to be subject to the general sources of mutation, such as radiation or chemicals. Second, if you were to impact them in any significant way, which the only time you can do that is during the early embryonic phase, it would actually be fatal to the organism and its development. So for this reason, it's very doubtful to get new epigenetic information. Now those are the three components that scientists have identified new body forms require. We saw that the odds of just getting one by itself is incredibly improbable. How much more all three together? You need all three arising together, making the new body plan. Just one or even two without the third doesn't give you a new functional body plan. You need all three. So that poses a problem. As explained by biochemist Paul Nelson, a biologist, if the only kind of mutations that can conceivably produce enough morphological change to alter whole body plans never causes beneficial inheritable changes, then it is difficult to see how mutation and selection could ever produce new body plans in the first place. Thus, life seems to be isolated within major kinds. And that's what we've learned from contemporary science, molecular biology, genetics, and epigenetics. So the question is, knowing that, what is the picture we have of life, assuming the fossil record and our contemporary science? There are two options. First is the bottom-up option. Life starts simply and then branches out from there. In other words, we start with species, and then from that species we'll get different genuses and families and orders and so on up until phyla, the major animal types. The second picture is a top-down picture where you have major types from the start. In other words, you start with different phylas already in place, and then those phylas diversify within themselves: genuses, families, orders, classes, and ultimately classes, orders, families, genuses, and ultimately species. Now, those are two very different pictures. What do we find in the fossil record? We find, as explained by biochemist Roger Lewin, who was highlighted in the journal Science, several possible patterns exist for the establishment of higher taxa, the two most obvious of which are the bottom-up and the top-down approaches. In the first, evolutionary novelties emerge bit by bit. The Cambrian explosion appears to conform to the second pattern, the top-down effect. Or as explained by paleontologists Douglas Irwin, James Valentine, and Jack Sapkowski, the fossil record suggests that the major pulse of diversification of phyla occurs before that of classes, classes before that of orders, orders before that of families, and so on. The higher taxa do not seem to have diverged through an accumulation of lower taxa. As explained by paleontologist Richard Fortey, the beginning of the Cambria saw the sudden appearance in the fossil record of almost all main types of animals, phyla, that still dominate the biota today. And lastly, as Charles Darwin himself admits, the abrupt manner in which whole groups of species suddenly appear in certain formations has been urged by several paleontologists, for instance by Agassiz, Pictet, and Sedgwick, as a fatal objection to the belief in the transmutation of species. If numerous species belong to the same genera or families have really started into life all at once, the fact would be fatal to the theory of descent with slow modification through natural processes. This is a major problem to the one who holds the belief that life arises by merely naturalistic processes. So as I kind of wrap up and lead us to the conclusions, what have we found so far in this argument? So here are some problems that we found with the neo-Darwinian view of life. First, the sudden appearance of complex life forms, the absence of ancestral fossils, completely new forms and body plans, isolation of those body plans, and lastly, major difference from the start. That's the picture of life that we found. So the question is, if that's the picture of life, that's what happened, what explains it? In order to answer that, you need to recognize that life has both invention and information. Now, what do I mean by invention? I mean the specific arrangement of specific parts into higher complex structures resulting in a function. Now, you might look at a pile of sand. That's not going to be what we understand as invention. You can cut it in half, still have a pile of sand. Nothing really happened, right? No particular arrangement, no particular parts, no particular function. A circuit board, on the other hand, if you cut that in half, it's no longer full. It doesn't accomplish its purpose, right? It's the specific arrangement of specific parts collected into a higher complex structure for a function. That's what I mean by invention. Now, what we find is that life is an example of that latter, the invention. If components for body plans are not present or assembled properly, the invention is not functional. We need all parts to be present and arranged just right. Specificity is key. That word's going to be huge when understanding invention. Now, some have argued that life is irreducibly complex, and that's another conversation. I'm just going to brush over it, but basically, they argue that all these specific parts need to be present and arranged just right from the start. They don't progress and eventually get there. In order to have life functional and being able to survive from the start, you need all these pieces just right and organized properly. An example given is a mousetrap. You know, there are 5 parts. If you take out one, or if it's not put together just right, it won't function. As Charles Darwin states, If it could be demonstrated that any complex organism existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. Now, some have argued, such as biochemist Michael Behe, that such structures exist. Here are a couple examples. That's another conversation on the side that could be dove into. I just want to talk about what is the simplest examples of life that we find, and what does that tell us about the complexity and how it must have been that complex from the start. Now, it's been determined what the minimally complex cell would require, and it would require about 250 proteins bonded just right, each composed of about 150 amino acids. Now, what is the probability of getting one of these simplest living cells from a prebiotic soup? It's been calculated to be just one chance in 10 to the 41,000th power. What does that look like? According to molecular biologists, if all the events in history were devoted to simply trying to throw stuff together and result in this simplest cell, we would have only 1 out of a trillion chances. And that's just to have a 50% chance. That's not even to have a good chance of getting it. For the simplest living cell, even in the history of the universe itself, seems unlikely. There's simply no such thing as simple life. See, we can only assume that life arose in the absence of intelligence when we assume it's simple. What we find is that there's no such thing as simple life. Even the simplest cell by itself is not simple. It's an example of stunning invention. As explained by biochemist Michael Denton, the complexity of the simplest known type of cell is so great that it is impossible to accept that such an object could have been thrown together suddenly by some kind of freakish, vastly improbable event. Such an occurrence would be indistinguishable from miracle. And he goes on to say, molecular biology has shown that even the simplest of all living systems on Earth today, bacterial cells, are exceedingly complex objects. Although the tiniest bacterial cells are incredibly small, weighing less than 10 to 12 GMS, each is in effect a veritable micro-miniaturized factory containing thousands of exquisitely designed pieces of intricate molecular machinery, made up altogether of 100,000 million atoms, far more complicated than any machine built by man, and absolutely without parallel in the non-living world. So we see that there's abundant and impressive invention within life. But let's wrap up talking about information. According to Professor of Zoology at Oxford University, an atheist himself, he said that life, even at its simplest, the simplest cell contains the equivalent amount of information, specified complexity, which we talked about earlier, as 30 complete sets of the Encyclopedia Britannica. The simplest cell. How about the simplest organism, the amoeba? He said that the amoeba has the equivalent amount of information, specified complexity, as 1,000 complete sets of the Encyclopedia Britannica. Once again, there's no such thing as simple life. There's no such thing as life that is not information rich. As explained by Bill Gates, the founder of Microsoft, DNA is like a computer program, but far more advanced than any software ever created. Life, at its simplest, is the greatest example of invention and information on the planet, unparalleled in our own human invention. So, as we wrap up this argument, we ask, where does invention and information come from? Now, because invention and information are examples of specified complexity, you can equally ask, where does specified complexity come from? And the answer is intelligence. Our repeated experience confirms that only intelligence is capable of doing this for one simple reason. Organization does not happen by accident. We talked about entropy earlier. The universe is such that it becomes more disordered over time, not more ordered and organized. Now, organization, and we saw that these coordinated changes directed towards a specific function and purpose requires intentionality and purpose. Such things only exist within a mind. And that's why our repeated experience concludes that only intelligence is capable of such things. The question is, can we take that seriously? And the answer is yes. As we wrap up today's presentation on evidence for God, it can be taken seriously. In fact, we assume our repeated experience and the kind of thinking that detectives use. They ask first, what is capable of causing effect? What are the potential suspects? What is capable and strong enough to be able to do it? Second, what is present in order to be able to be there and actually do it? The suspect that was present is likely the one who committed the crime. Well, it's the same when it comes to this argument for intelligent design. We first must ask, what is capable? Now, here's the interesting thing. If only one suspect is capable, you're justified in assuming it was present. What's that even look like? We see that if we are exploring Mars, let's imagine that we happen across a drone or some type of machinery. What are we going to conclude? Did it come together by natural processes, sandstorm? I don't know. No, we'd be justified in concluding that it was the result of intelligence being present. Even though we don't know of intelligence being present on Mars, we'd be justified in saying that at some point in the past, intelligence was here to be able to create this type of invention. Because specified complexity like this only comes from intelligence. Now, we saw that there's invention and information within life. So we ask, what is capable of producing such things? Our experience confirms that only intelligence can do that. So we are justified in concluding that intelligence, in some shape or form, was present at the arrival of life to create this invention and information that was there from the start. As we've seen, even the simplest organisms have stunning amounts of invention and information. So that's the argument from intelligent design. We see that life is a great example of invention and information, and such things only come from the mind of intelligence. As explained by molecular biologist Douglas Axe, the reason we perceive purpose in inventions is precisely the reason they can't occur by accident. They exhibit an organized functional coherence that can only come from deliberate, intelligent action. I can only see these ingenious creeping, climbing, swimming, soaring, blooming, burrowing, luring, lunging, spinning, sporulating, fleeing, and fighting inventions as having come from the mind of God. To me, nothing else makes any sense. Or stated by biochemist Michael Behe, life on Earth at its most fundamental level and its most critical components is the product of intelligent activity. The conclusion of intelligent design flows naturally from the data itself, not from sacred books or sectarian beliefs. And lastly, as explained by philosopher of science Stephen C. Meyer, standard materialistic evolutionary theories have failed to identify an adequate mechanism or cause for precisely those attributes of living forms that we know from experience only intelligence, conscious, rational activity, is capable of producing. So we see that purpose is indeed found within life. It arose rapidly, already complex and diverse. We see that the invention and information found within life is far more complicated than any human engineering or coding ever present. Far more incredible and stunning. So going back to this whole presentation, what kind of universe are we living in? Are we living in a universe with a God or without a God, like Richard Dawkins talked about? What best explains it? What is the product of the greatest conceivable mind, and what is some sort of freak accident? We see that to disbelieve in God is to believe that the universe came from nothing, order came from disorder, life from non-life, invention from blind processes, information from chaos. We live in blind, pitiless indifference with no rhyme or reason to life. If that's the view you take, those are the implications into what human life is, why we're here, how we ought to live, and where we're going. But everyone has to choose. We each have to choose how we're going to answer that question. Does God exist? No one's going to force you to come to any particular conclusion. You have to choose for yourself. But I think there seems to be abundant and remarkable reasons for belief for the one who's open to it. Hebrews 11:6, as we heard earlier, says, "And without faith it is impossible to please him. For whoever would draw near to God must believe that he exists and that he rewards those who seek him." So perhaps there is great reason for belief indeed.